Hi friends, welcome back to the JK Biology, and today I will be described about the eukaryotic transcriptions and how it differ from the prokaryotic transcriptions and which polymerases involved in the eukaryotes and how it differ from the prokaryotic RNA polymerases. Okay, so there is the three types of RNA polymerase that is involved to the eukaryotic transcription, that is RNA polymerase first, second and third. But in prokaryotes only single RNA polymerase is required, that is RNA polymerase third, that contain alpha, 2 beta, beta dash and omega subunits, that is the core subunits for activations of the transcription, and there is another sigma factors that start the transcriptions and also help to recognize the RNA polymerase. Okay, but there is the different RNA polymerases, that is RNA polymerase thirds, first, second and thirds involved in eukaryotic transcriptions. First, I will describe about the alpha amantine, that is the most frequently questions that asked in the examinations. Alpha amantine is a poison that is obtained from Amanita phylloides. Okay, that is the fungus. Inhibit totally RNA polymerase seconds, that's inhibit totally on RNA polymerase seconds, but that becomes unaffected. RNA polymerase first becomes unaffected by the alpha amantine, that is resistant, but there is moderately affected, okay, affected by the alpha amantines. Okay, so there is the 12 subunits, nearly about 12 subunits of RNA polymerase eukaryotes found, but there is the 6 subunits, 5 alpha 2 beta beta dash and so omega, but there is 12 subunits more than prokaryotic subunits. Okay, so there is the in RNA polymerase seconds the largest subunits contain RNA polymerase second, that is second, contain carboxy terminal domain, that's act as an array of binding to the protein, that is the site for array, that is site for array of proteins to the binding in the C carboxy terminal domain. Okay, and also act as a substrate for different kinases, also act as substrate for kinases. Okay, and also another first, that is second, for binding site, this is the site for binding array of protein, this is the site for binding array of protein. So, and also RNA polymerase in the mitochondria of eukaryotes, RNA polymerase, all RNA polymerase gene is encoded in the by the nuclear genomes. Okay, and so in the chloroplast, there is two types of RNA polymerases found, that is one encoded by the nuclear genome and another encoded by the self of chloroplast. Okay, but in the self of chloroplast, RNA polymerase resembles to the bacterial RNA polymerases, alpha 2 beta beta dash, but the sigma subunit or sigma factors that is encoded by the nuclear genomes, except all core proteins, core subunits, alpha 2 beta beta dash and omega is encoded by the chloroplast genome of, I am saying that there is two types of RNA polymerases, one is encoded by the nuclear genomes and another is chloroplast. Which is encoded by the chloroplast, that resembles to the bacterial, except sigma factors that is encoded into the nuclear genome, but another RNA polymerases that's totally encoded by the nuclear genome of the eukaryotes. Okay, so this was the chloroplast and mitochondria. Mitochondria also resembles from the prokaryotic RNA polymerases. Okay, then come to the point, that's where RNA polymerase binds, how it start the transcriptions. RNA polymerase binding sites have a specific sites for binding on the DNA, that is called promoter. Okay, promoter is a specific sequences where RNA polymerase binds, that contain the consensus sequences Okay, so RNA pole first have another types of the promoters, seconds have another types, and thirds also have another types for transcribing of RNA. But all these transcribe different types of RNAs. Okay, first promoters. I am describing about the first. Okay, this is the promoter. +1 start sites. Okay, and if you go to the right of the DNA sequences, this is the DNA sequences goes to the right, then there is called downstreams, that is +1, +2, +3, and so on. If you go to the left sides, that means upstream, that becomes -1, -2, -3, and so on. Okay, here this is the type first promoters, where that is found initiator sequences, that's become AT rich. But there is core promoters. Okay, there is core promoters, that's become GC rich. Okay, and another that is called upstream control element is found from the start site -100 base pair long to -180 base pair long. If RNA polymerase first binds, that's transcribed to the rRNA. Okay, rRNA in which include 5.8S and 18S and 28S. Type first RNA polymerase transcribed 5.8, 18, and 28S. This was the sequence of RNA polymerase first type of the promoters. Okay, and come to the RNA polymerase third. First I am describing about the RNA polymerase third promoter, that is third pole third promoters. Okay, pole third polymerase contain three types of the promoters, that is type first, second, and third. Point out that is RNA polymerase third contains three types of promoter, type first, type second, type third. Okay, so there is the first promoter. What is first promoter of RNA polymerase third? +1. Okay, upstream and downstream, there is the specific sequences, that is called A box and this is called C box. Okay, this is C box, A box, and that's transcribed 5S RNA. That's transcribed 5S RNAs. Okay, this was type first promoters of RNA polymerase third. Okay, type second promoter. Type second promoter of RNA polymerase third. There is +1, also contains box but differ from first. Okay, that is A box and that is called B box. Okay, from the start site, this is the DNA sequences, that's transcribed tRNAs. That's transcribed tRNA, and another is type third promoter. Type third promoters. Okay, this was type second promoters, this was type third promoters. Okay, then there is +1 and here is the Tata box found -30 sequences, this is called, and another is found proximal sequence element and here is digital sequence element. Okay, proximal sequence element is found -44 to -66 upstream sites of the base pair sequences, and distal sequence elements is found -214 to -244 distance from the start sites. Distal sequence elements. Okay, and this is the +1. It transcribed the U6 snRNA. U6 snRNA type 3rd promoters. Okay, so this was the sequence and the promoter of RNA polymerase thirds. Now promoter for RNA polymerase second, then promoter for RNA polymerase seconds. Okay, this is +1 and here is the Tata box. Okay, Tata box, that is the conserve. What is the means of conserve sequences? Consensus sequences, sorry. What is the means of consensus sequences? There is the first TACGT, second GTAGCA, okay, and here is TAGCT. That is the three types of the DNA sequences in which one is common. A is common, that is called consensus sequences. That is, A is consensus sequences in 3 DNA sequences. Okay, so there is the Tata box, that's become long length of DNA sequences in which TA is the consensus sequences where RNA polymerase binds. Okay, and another that is initiator INR. Initiator, that's become AT rich, also AT rich, and here also act as cis acting element, that's act as regulatory, that's either increases transcription or decreases transcription. That means regulatory sequences is found in the RNA polymerase second. This is type RNA pol second. Okay, so that's either inhibit the specific transcription proteins or transcription RNA or increases the transcription of RNA. That is, sequences found is here GC box and CART box, that act as regulatory cis element for RNA polymerase second. Okay, so it transcribed the mRNA. Okay, it transcribed mRNA. Initiator sequences becomes AT rich and especially it's become pyrimidine rich. Pyrimidine rich sequences. Okay, this become pyrimidine rich sequences. CART box, GC box. This is the part of RNA polymerase second. You have seen that these all promoters differ from the prokaryotic sequences, and that is the Tata box, GC box, CART box is the part of RNA polymerase second of eukaryotic genes. Okay, so there is also sometimes downstream some elements is found. Points out, CART box, GC box is orientation independent. That does not depends where is the orientation 5 to 3 or 3 to 5. It's become transcribed. So it, because it acts as regulatory elements, it can also found to the downstream, either it can also found to the upstream. So there is orientation independent. But the Tata box, that's become +1 start site initiator sequence. Around the +1 start site initiator sequences is found. That's become pyrimidine rich. But there is a data box where transcription factors first. That's general transcription factors. In further lectures I will be described about the general transcription factor and other transcription factors. Here data box where general transcription factors binds and start the transcriptions. Okay. So this was the description about the promoters of RNA polymerases, and hope this lectures would be beneficial for all viewers. And if you like this lecture, then please click on subscribe button and also share for your friends. Thank you.